Hello and welcome to Bone Up, the podcast all about bones, how we make them, why we break them, and if we fully understand them. I'm David Armstrong.
Hi, and I'm Richie Abel. And over this series, we're going to be exploring osteoporosis, bones, what we know and what we're yet to discover. And we hope you will join us on the journey.
So, for anyone keen to learn more about our infrastructure of calcified collagen, this is Bone Up.
Welcome back, listeners, to part 2 of ECTS Live 2023. We spent some time, David and I, running around at the poster exhibition and talking to some of the young researchers at the conference. And here they are now. We're here with our next guest.
Okay.
Nice to meet you. Could you
Me
tell us
too.
your name and where you're from?
Yes, Zora Bruchemla from Paris, Bioscar Lab. The directress is Martine Cohen-Solal. You must know her.
And are you doing a PhD there?
Yes, I'm doing a PhD. I'm third year PhD.
Wow.
And I'm working mostly on osteoarthritis and cartilage.
And you're presenting a poster about it at the
Yes,
conference?
I'm presenting a poster, and I have a commented poster with a short presentation of 3 minutes. So my poster is about Lean 28A that induces chondrocyte energy metabolism through SDHA inhibition.
And why is that important?
It's really important because we hope therapeutics, then it will be a therapeutic strategy for osteoarthritis. So the main protein is Lean 28A, and it's really involved in cartilage regeneration.
So for the patients I see at clinics, because we have lots of patients listen to the podcast,
Yeah.
people, when they think of osteoarthritis, they say to me, my bones are rubbing together. But actually, long before that, it's the cartilage that is the problem. It's the cartilage that wears out and gives way. And you're working on some way to regenerate the cartilage. Is that right? So that sounds exciting.
Exactly.
Where do you think this would go then? Is this a tablet someone will take? Is it an injection they'll get? Or is it something a surgeon would do at an operation, putting something into the cartilage?
I think that it will be mostly local injection,
Okay.
or maybe using new technology. Now we have the particles where we can target specifically tissue with these nanoparticles that I
Right.
heard from Congress and everything industry. So I think it would be really interesting to think about that after. Now we are just looking at the mechanism of the protein, how it can regenerate cartilage, what are the pathways involved.
So the idea we would inject something into a worn piece of
Yes,
cartilage
in
to
the
somehow
knee
stimulate that to rebuild.
Yes, in the joint.
Yeah, because I've always thought we sometimes see patients who maybe one side of their knee is very badly worn, the other side's not so worn, but it's just the cartilage is worn, and the
Yes.
orthopedic surgeon will often say, come back in 5 years when I need to replace the
Prosthesis,
knee.
yes.
So this is really exciting that this is something we can actually prevent the need for knee replacement.
And we have some data on human, because we have access to cartilage, section cartilage from human in the lab, because we have the hospital just near, and we perform some experiment, and it's really concluent. So I hope that one day I will see my protein as a therapeutic strategy.
You will see it in someone's knee.
I hope, not in mine.
Not in yours, hopefully not in yours.
Thank you very much for talking to us today,
You're
Sarah.
welcome.
That's interesting.
Thank you.
That's been lovely, thank you.
Thank you very much.
Now we have Ophelia from Toulouse. Welcome.
Well, thank you.
Which institute are you from?
I'm from the Pharmacological and Structural Biology Institute in Toulouse, so IPBS, in Toulouse, France.
And you have a talk here at the conference?
Yes, I had a talk this morning. It was about the role of muesin in osteoclastogenesis.
And what were the findings of your research?
I found that muesin, which is a membrane to cortex protein that binds the plasma membrane, and also the effectin, had a regulatory role or inhibitory role in osteoclastogenesis. at two levels. The first one in the fusion of osteoclasts, and the second one in the degradation of the bone degradation ability of those osteoclasts.
So osteoclasts then are a multinucleated cell, and
Yes.
I suppose what you've been able to do is understand how these cells come together and join to make the osteoclast.
Yes, actually they are doing a really nice process that is the formation of membranous protrusion called also tunneling nanotubes. And what I show is that by depleting muesin, we increase the formation of tunneling nanotubes. So we think that muesin can regulate this formation and thus the fusion of those multinucleated cells.
Now, my patients are interested in osteoclasts because I explain to them osteoclasts are the cells that stick to the surface of their bones and tunnel holes in their bones and lead to osteoporosis. And we often say after the menopause, these cells work much faster and you lose bone. So your work is on controlling how the osteoclasts are formed and also on how well they work. Is that correct?
That is correct.
And presumably then, if you understand which proteins control that process, if we were able to fiddle about with those proteins, we'd be able to maybe make the osteoclast less active?
Yeah, we would hope so. So in order for this protein to work, it has to be activated. And the thing is, this protein is expressed by a very large amount of cells in our body. And if we deplete it or control it, maybe it will have second effect.
Mm-hmm.
So what we are going to do is to go upstream to see which effectors can modulate this activity. And in those effectors, we want to identify one which is particularly expressed by osteoclasts.
So you have to find a step in the pathway then that can lead to an effect on the osteoclasts,
Yes.
but without having unintended consequences elsewhere in the body.
Yes.
Do you think that's going to be possible, or is the process too complex?
Actually, we started to identify some of these molecules in the pathway. However, the thing is, each of the three molecules that I identified in the pathway seems to be expressed by another large amount of cells. So, for now, we are in a dead end.
We're still at the stage, I think, with a lot of medicines where we switch something off completely in the body or switch something on completely in the body. It interests me because you're looking in such a detailed way of proteins that control other proteins that control osteoclasts. There's a possibility here of being much more subtle in what we do if we could eventually develop some drug based on this.
We would like to, actually, yeah.
I think everybody would like you to. Thank you very much for coming to talk to us. That was really interesting.
Thank you.
Thank you
That's
to
great.
you too.
Thank you.
So, sorry.
Another guest, we're delighted to speak to Annelies Schmidt from the Netherlands.
Yes.
You work with Kevin, who we spoke to just earlier.
Yes, that's right.
And tell us what you do. You work in the laboratory, is that right?
Yes, I'm actually the only one of our group that is in the laboratory because the other ones are all clinicians. So I'm studying the importance of circadian rhythm on bone health. And actually I'm specifically focused on what is the communicator of central rhythm to bone health. Previous research has identified glucocorticoids as an important cue for bone health and also for rhythm. And now I'm actually investigating in how does this actually work and how can we make use of this for in the clinic. So
And circadian rhythm is the hormones in the body rising and falling over 24 hours and how that affects what
Yes.
goes on in the body. So what particular aspects are you looking at then?
Yes, so first we established that circadian rhythm is actually important for bone health. So we performed a study in which we disturbed the light-dark rhythm and then we saw an osteoporotic phenotype, so bone became porous and breaks more easily. And then we looked at multiple hormones to see what hormone might correlate with circadian rhythm and with bone rhythm, and then we identified glucocorticoid rhythm as an important regulator, which is also supported by other research, of course. And what we do is we flatten the glucocorticoid rhythm in mice, so they do not have a diurnal variation anymore, and then we observe the effect on bone parameters, so on bone structure, strength, and other outcomes.
This is incredible research. This is really interesting. So the experiments that you did when you did the light and dark, was that on bone tissue samples or animals? What did you use?
Yeah, so it was actually a PhD student who was working on this project previously, and what she did was, it was a study in mice, and it's called a jet lag model. So you put mice on a continuous jet lag, so they cannot. They cannot keep up with it.
And then fly them around the world, is that right? Yeah.
No, you just switch. I think maybe every 3 days or something, you switch their rhythm.
Light and dark.
Yeah. And then what you look at is the body composition of mice, just some general factors, so lean mass, fat mass, body weight, and then you go deeper. So you look at the bone tissue structure, the mass, those kind, and also the strength. And then you also go a bit deeper and you try to look at how bone genes are regulated and whether the regulation of genes is also affected. So you look at multiple levels.
This project is mind-boggling. That's incredible. So you can go all the way from the genes, I guess, the gene expression, the effect of the changes in circadian rhythms, right through to the strength of the bone at the end.
Yes.
And if somebody has a bad circadian rhythm, if you're messing around with the sleep, does the bone strength go down?
Yes, actually, you see people who work in shifts, for example, nurses, they have an increased osteoporotic risk. So this is really, it's really clear that not or people that, for example, pilots or...
So we need to add that to lifestyle factors. So when we talk about food and exercise, we now think about environmental factors as well, pollution in the atmosphere. So sleep deprivation and altered circadian rhythm, we should be adding that to our list of things we ask patients about. Yes. That is really interesting.
Yes, and sometimes I get a question like, how does it work in blind people? Because they have no light-dark rhythm, but actually you can sustain your rhythm with other ways. So there are a lot of things that regulate your rhythm, so light-dark cycles, sleep rhythm, exercise patterns, foods intake. So you can always try to work around what patterns work best for you and try to keep your rhythm as good as possible.
Sounds like conference attendance might be a risk factor for osteoporosis. It's long days and I think we're all lacking sleep.
Yeah.
And Elisa, thank you very much. That was wonderful.
Really good. Thank you very much.
Okay, well done.
There's even some round of applause
as well. Yeah.
We're here with Scott Dillon from Cambridge University. Welcome to BoneUP.
Oh, thank you so much.
How have you found the ECTS conference this year?
Yeah, it's been really great so far. We've had the Bone Research Society conference appended onto the front of ECTS this year. I'm not a usual ECTS goer, so having both conferences together this year has been a great opportunity to sort of meet some people that I would have never crossed paths with otherwise, and I've already had some great discussions over the last couple of days at ECTS, so it's been great.
Nice to hear another Northern Ireland accent. Tell us a little about the work you presented earlier in the conference then at the BRS.
Yeah, so I presented an interesting talk at the Rare Bone Disease Workshop this year at the BRS. Again, it was a first for me. I don't usually go to the Rare Bone Disease Workshop because I'm a sort of cell biologist turned amateur chemist. But this year we had some exciting work with Ken Poole at Addenbrooke's, where we're doing some combined sort of clinical investigations of one of his patients and then doing some chemistry on those samples. So it was really interesting to get a more clinical perspective on the sort of work that we're doing at the Rare Bone.
We've often talked about rare bone diseases and how we can learn things about common bone diseases from looking at rare bone diseases. Is there anything stands out from your work in that sense?
Yeah, well, that work from Ken has been really interesting because the patient that we're mostly focusing on in that work is a rare form of hypophosphatasia, where she has compound heterozygous mutations in tissue non-specific alkaline phosphatase, which is the causative, sort of most common causative mechanism of that disease. But what we've really been able to do is actually compare that with other disorders of hypomineralization, so in this case specifically osteomalacia, caused by various factors, and we've seen some really interesting differences in the sort of chemistry of the mineral at the atomic scale using nuclear magnetic resonance in this case. So that's been really fascinating to compare sort of similar phenotypes with distinct causes and find actual differences in the biology.
Yeah. So much we're hearing about now is quality of bone rather than quantity of bone, which is particularly relevant, I suppose, to osteomalacia and all diseases of hypomineralization of bone.
Yeah, that's right. And we had a brilliant talk from Duncan Bassett at the Bone Research Society, who's really get going on this bone quality measurement and finding these genetic determinants of bone quality, which was also really fascinating.
Yeah, I hope that over the next sort of 5 years we're going to see a burgeoning increase in the amount of bone quality research. For a long time people have been really focused, really obsessed with bone quantity, I guess, because mostly in clinical practice bone diseases are targeted through DEXA-based BMD. Now we've got all these wonderful new technologies and a whole crop of people who are maybe chemists and other backgrounds coming into bone biology. I think maybe we can start to make some real headway.
Yeah, I think that's right. I mean, in this case in particular, the whole reason that Ken, who's the clinician in this case, came to us was because he found this patient had huge accumulation of sort of unmineralized osteoid on her tuberculae, but simultaneously massively high bone mineral density. So he sort of came to us as basic scientists and chemists and said, how can this possibly be? And can we use some of your physical chemistry techniques to try and work out what's happening down at the nanoscale?
I think Ken Paul's a really good example of a clinician who really thinks outside the box and is very good at engaging scientists from all sorts of disciplines to go and help and support him to improve his patient care.
Yeah, that's right. And it helps that we're sort of quite a close-knit community in Cambridge because there's not very many sort of bone researchers, musculoskeletal researchers in Cambridge, so we all know each other quite well.
Oh, wonderful.
It's one of the things we always try to do here is promote links between science and clinician and the patient, which it sounds as if you're right, and certainly working with Ken, you're right in the middle of that, which is great.
Yeah. I mean, there are challenges for us as basic researchers. We generally don't understand the ethics process is completely alien to us, but luckily, in collaboration with Ken, we've been able to navigate that really effectively.
Great.
Wonderful. Thank you for talking to us.
Thank you.
Thanks, Scott.
We're lucky now to have Paul Rothwell with us. Welcome.
All right, thank you very much for having me.
Are you enjoying the conference?
Yeah, it's been great. It's been lovely to present some of my sort of early research here from my part-time PhD, but also to be able to go to so many different lectures on sort of different subjects. I learned so much, not just on my research, but on other things as well. And it's great to have two conferences running one after the other as well, the BRS and then ECTS, to be able to come to both.
Could you tell us about your PhD? What's the work on?
Yes, I'm currently studying a part-time PhD, and I'm looking at the whether diabetes and arthritis have interlink really, whether that the effect of diabetes on the blood vessels is actually causing arthritis. We know that the two coexist, and as people get older, they get these diseases as well. But I'm specifically trying to look at if the blood vessels are damaged, mainly looking at the big blood vessels, so down the back of the knee, the popliteal artery, and seeing if there's a smaller lumen in there as the artery gets thicker, if that's having an actual effect on the arthritis in the knee as well. I'm also trying to take into account the person's BMI as well, because obviously people will say that it's just the weight that is causing the arthritis in the knee. So I'm looking at healthy BMI individuals to see if it is the arteries that's causing that.
That's very nicely explained. When I was a lad and learning about diabetes, we were taught that the side effect or the effects on the vasculature were divided into the microvascular and macro, in other words, big vessels and small vessels. And I think we always imagined that the effects on bones might be small vessel disease, but you're looking actually at the big vessels?
Yeah, just really in terms of the nutrients that are able to get towards the knee itself. We know that cartilage is avascular, so we don't actually get blood vessels heading towards there, but through that diffusion, the blood vessels are getting in towards the bone itself, into these tiny little blood vessels, and I'm seeing whether the damage to the bigger blood vessels is a marker of damage to the smaller blood vessels. So hopefully, if we're able to see a relationship, which I have done in the early stages of my research, that we can almost intervene before it gets too bad, and we can try and encourage patients with diabetes to actually look after their things like their diet and things like that, and also take the medication that they're meant to be taking in order to prevent things like arthritis happening later on in life.
Yeah, we've talked a lot about the connection between bones and blood vessels over the last couple of series, and as time goes on, the more and more interconnections we see, it's fascinating. You had a slightly unusual career path compared to some of us here. Tell us a little just about how you got to where you are now.
Yes, after leaving college, I wasn't too sure what degree I wanted to do or what job I wanted to do, and I luckily got a job at the University of Liverpool as a research technician. I was very fortunate to work in Professor Jim Gallagher's lab, as well as lots of other labs within the anatomy department there. Learned a broad range of techniques, which is helping me in my future, what's going to be my future career. And then through some changes in the department, I also studied a part-time degree whilst I was doing that in biomedical sciences. And then through some changes in the department, I then transferred job into sort of our human anatomy resource centre, where I was a teaching technician. Fell in love with teaching and wanted to do that as my career, really. So now I lecture to anatomy students, whether they're sort of medical students coming to learn anatomy, allied health, dentists, or just pure anatomy students. And now study a part-time PhD alongside that.
Yeah, it's inspiring to hear that you've done those both your degrees as part-time, and yet here you are successfully presenting at a conference. There'll be a lot of people will be, excited and inspired to hear that. That's a career option.
Yeah, I think it's really important for me personally to really love what I'm doing as a job. So that's why I didn't sort of jump into a specific subject at the start, and really, sort of almost learn to love my job. And I think because I'm doing something I feel passionate about, I think the research output as well will be much better as well, because I'm really invested into what I'm actually doing. I really love it, and quite addicted to the research that I'm doing at the moment.
That's really, really good to hear that.
That's wonderful and very well explained. I concur with David. Thank you for your time.
Okay, thank you very much. Cheers.
Great to talk to you.
Hi, welcome to ECTS. Could you tell us your name and where you're from?
My name is Nikola Stokovic. I'm from Croatia. I'm currently working at the University of Zagreb School of Medicine in the Laboratory for Mineralized Tissues and the Department of Anatomy.
Oh, wonderful. And you're presenting a poster at the conference. What's it about?
Yes, I'm presenting a poster on the new bone regeneration device, which is called OsteoGrow-C. It is composed of bone morphogenetic protein 6, which is delivered within autologous blood coagulum as a BMP carrier with synthetic bioceramics as a compressive-resistant matrix. And in this study, we tested this novel bone regeneration device in posterolateral spinal fusion model in sheep.
Wow. And big question, I suppose, did it work?
Yes, it worked. So it achieved spinal fusion approximately 7 weeks after implantation. And in this specific study, we evaluated the long-term outcome after 40 weeks. So it is almost 1 year, and the spinal fusion was sustained, and therefore this osteoinductive device is a promising solution for posterolateral spinal fusion, but also other clinical indications.
That was going to be my question, because I'm a clinician and I see patients in the clinic every day, and they're maybe going to be listening to this and wondering, what does this mean for me? This is a device which helps build bone. Thinking of someone who's not a scientist listening to this, so lots of the drugs I give people stop bone loss. This builds new bone.
Yes, it builds a new bone, and it can be used for achieving bone on sites like spinal fusion, so between the transverse processes or between the vertebral bodies, but also it can be used for rebridgement of segmental defects and indications like
And for my patients listening to this will be an operation. They will having something placed into the bone that will sort of grow and expand and strengthen it, or an injection, or how do you see it working out clinically?
It should be placed during the surgical procedure, and this is osteogrowth C. This is one of the specific form of osteogrowth, because osteogrowth is a device containing BMP6 within autologous blood coagulum, and it is already in clinical trials in different indications like posterolateral spinal fusion and the trial with the high tibial osteotomy and distal radial fracture has already been done.
And you see this primarily as a treatment for spinal problems then? Or is it just you happen to be looking in the spinal area in your models?
This is one possible indication, but in my opinion, the best indication for osteogrowth C are spinal fusions, so different. It can be anterior lumbar interbody fusion, posterolateral spinal fusion, and also regeneration of long bone segmental defects.
Wow, a lot of uses.
That's very, very exciting. We look forward eventually to seeing this in the clinic. That's my take on all these things. So that's great. Thank you very much for speaking to us.
Thank you very much. We're here with Guido and Filippo from Italy. Welcome. How are you both doing?
Nice to meet you. Well, good. This is our first time here at
it's a good experience.
ECTS. Nice experience, yeah.
I'm sorry the weather isn't better for you.
we know that we expect like this, yeah.
You expect that. That's good. That's good. You've been doing some research in an area that is of particular interest to us. We've mentioned it before in the podcast, and that's bone quality, particularly with respect to diabetes. Could you tell us a little what you're going to present on later today?
Yeah, we have stratified the risk of fracture in diabetic patients, not only evaluating bone parts, so with items such as DEXA and FRAX questionnaire, and previous fracture or incident fracture, but we evaluate even the diabetic status of the patient. So the presence of complication of the disease, the presence of a long duration of the disease, or a bad glycemic control. Over the use of certain drugs that can alterate the bone homeostasis. And following this algorithm, we found that not only BMD could be useful in this patient, but also these other items could be useful, more useful of BMD, because as we know, BMD could not be so reliable in diabetic patients because usually they have higher values than postmenopausal osteoporosis, for example.
And so you're trying to finesse this a little, because at the moment, if we're calculating a FRAX score, we can sort of tick a box for diabetes, but you're looking at how long the diabetes has been there for, how good the glycemic control. You're trying to look inside the diabetic box, is that right?
We try to look in all the items of the patients. We are clinicians, so we try to look the patient has together of many things. And especially for diabetics who has some complication could affect kidney, eye, heart and vessels. How affect the vessels of these organs can affect both the vessels of the bone, for example.
And so the big question is, if you have worse, let's say, eye disease or kidney disease because of your diabetes, does that put you at higher fracture risk? Is that a marker?
In FRAX risk, FRAX questionnaire, we evaluate diabetes comparing it at rheumatoid arthritis or with the TBS correction. In fracture risk, we see that depends. In this case, we have found more incident fracture in patients with more than one complications of the diabetic disease and maybe with a long duration of disease. Not only for the complication like retinopathy that could be a problem for the visual of the patient and the falls, especially for the neuropathy for the falls, but even because the alteration of the vessels of these organs can be even the vessels of the bones. And we know that the alteration of the vessel could increase the cortical porosity of the bone, for example.
And is blood vessel disease and diabetes particularly linked with fracture risk, do you think?
Could be linked. Could be linked in the way that could alter, could damage the bone homeostasis.
This is fascinating research. It's amazing that the risk factors that you can see, the other effects of the diabetes, are also risk factors for osteoporosis and falls as well.
Yeah.
It'd be fascinating to follow up with you in the future and see how far you get.
Now the study that I present this afternoon is a retrospectively study. So we have a population and we see what happens in this population on the base of this risk factor and in the percentual of incident fracture that they present in the observation time. Now we are trying to look even in perspective, so we are trying to have the assessment in our outpatients that come to our clinic and see what happens.
Do you think we could see a day when we would recommend to someone, if they control their diabetes better, they would have less risk of fracturing?
Yeah. However, it could be useful to inform even the diabetic patients that how he goes to cardiologist once in a year and to oculist once in a year.
Yep.
Maybe he has to do an assessment of the bone health once a year.
Yeah. We should be looking at bone health as well as retina and kidneys and all the other things.
Especially,
we look at.
we see that maybe some outpatients that come to us, if postmenopausal women, they already have a BMD with DEXA, but for postmenopausal, not for diabetics. Especially for the men who doesn't have this link with the postmenopausal, could be an item more that they could be, even because we see that mortality of post-fracture in diabetic patients could be more high than in non-diabetic ones.
Yeah.
It's a really exciting holistic approach to osteoporosis care.
Yeah. Maybe it's because we are clinician in internal medicine, so we have a holistic approach to the subject, to the patients. We don't see the single things, but we try to look the holistic way.
That's really useful.
Yeah.
Thank you very much for speaking to us. That's been very exciting. Thank you.
Thank you.
Cheers. Listeners, we have a very, very special guest, scholar, gentleman, Professor Jim Gallagher.
Thanks very much, Richard. I don't know how I can follow that. I'm very good. I'm on a high because the meeting's gone really well. Mm-hmm. Well, both meetings, the ECTS and the PRS. It was a little bit tense, especially on Thursday when the wind was blowing and various people were telling me they couldn't come, but I think it's all gone really well.
And tell us what was your role in the conference?
So I'm the local organizer of the ECTS and of course of the BRS, and the conference has been planned since 2016. Various things got in the way.
So what that was.
The most planning, I guess, took place when I was BRS president and Anna Teti was president of ECTS. But it had been discussed before then. And then, of course, we both stepped down, but this was the date that was set. And
so you've been doing bone research for a long time.
Yeah.
And you've been involved with ECTS and BRS for a long time.
Yeah.
How have things changed, do you think?
Yeah. So my first ECTS meeting was in 1981 in Knokke in Belgium. And I think my first BRS was in 82 in London. It was Bone and Tooth Society then.
It was. I remember that.
It was a meeting that Alan Boyd organised in 82. Yeah. How have things changed? Well, both societies then were quite small. BATS, Bone and Tooth Society, used to be a one-day meeting. We had 2 a year. There was very little, if any, corporate sponsorship. ECTS had some corporate sponsorship, but not much. And then over the years, both societies got bigger and bigger. Became more attendees, more sponsorship, and went, Bone and Tooth Society became Bone Research Society, and then we started having 2-day meetings and then 2.5-day meetings.
And how has the research changed in that time?
Well, that's interesting. I was honoured to do the Dent lecture the other day for BRS, which is the award they give every year. And so I took it as a sort of career award and went back to when I was doing my PhD. And so it reminded me, in those days we made our posters with Letraset. I don't know if you've even heard of that, Richard, have you? No, look it up.
For the benefit of listeners, Richie looks really confused.
We would type our own manuscripts. I typed my own thesis. I used to look after my own rats. I used to make the diets for them. Everything was very different. Years later, when we started doing PCR, I went to B&Q and got the perspex and made the tank myself. And of course there was no web then, so we used to get current contents. And when we went to a meeting, so current contents was a thing that came out every week with the front cover of every biomed journal, and you see, you go through that, and then you heard the names of these famous scientists. And I remember going to my first meetings, and these chaps would come along. I mean, obviously I'd worked for Herbie Fleisch in Switzerland, and I came back to work with Graham Russell in Sheffield, but all sort of the giants of the field, Larry Reutz, John Termine, these people. And it was such a buzz. They'd come and look at your posters, and I think that's one of the things that we don't do very well now. I think young people make nice posters, and I don't think enough of they get enough attention from the seniors. I mean, the other thing that's
We've managed to catch quite a few senior people down at the posters and interview them. Yeah, you can see them going around.
Yeah, that's good, and I think we really should. We really need to focus on that, and I'm as guilty as everybody, because there's always something. At meetings these days, there's always something to do, so you don't always get to
I think posters are a really important part of the meeting. I think it's something we really lost over COVID, when we were told, you can flick through the posters online, but there's nothing like actually walking around and standing and talking to people and seeing things. I think that's probably one of the most important parts of the meeting, and we didn't realise that until we lost them over COVID.
Yeah, and you can have a big impact on a young person's career, I think. So I think lots of things have changed, Richard. I think it's a lot more difficult to get funding now. I mean, I don't want to be depress you, but I think I've lived through the halcyon days of research. It was much easier to get established in those days. It's still possible now, and you have to make good connections. You have to come to meetings, and you have to be you have to have tenacity, because if you're turned down for a grant or your publication is rejected, you just got to bounce back.
What would you say to a young person, young researcher, young clinician, maybe, to make them interested in bone and osteoporosis? When there's so many fields, and there's maybe, you mentioned grants, it's maybe easier to get grants if you're researching heart disease or cancer.
Yeah.
Why bone?
That's a very difficult question, David. I mean, I did my honours project when I was a BSc student on bone. I did my MSc project, and I did my PhD on bone, and I've been in bone all the time. And it's always it's been, I've loved every minute of it. Actually, I love coming to meetings, but I also love being in the lab, and I love looking through the data. And I can think of a few occasions over my career where I mentioned this the other day in the Dent lecture, when we found that human bone cells were making osteocalcin. We were by the gamma counter watching the results come off, and it's still etched in my memory. So, it's the buzz really for me, and I hope. I mean, if people haven't got that, then they're probably in the wrong career. Yeah. But I mean, I think research, basic and clinical, is such a wonderful thing. And I mean, the last few years of my career, I worked on this rare disease, alkaptonuria, and that was a great story, because we actually found a therapy for that.
Yeah.
I think the challenges, there are different challenges for young clinical researchers, but I hope we can solve all those, and that we can keep good people. And BRS, our mission is to bring together clinical and basic research, and we're still working.
It works well. I think the great thing about the BRS and the ECTS is that there's lots of new investigators who get presentations. If you do bone research in the UK, almost everyone has their first talk at the BRS. It's where
Yeah.
it's where you meet the community.
Yeah.
You get over the first presentation you have to do, get over your nerves, become one of the people, and start making friends. It's really wonderful.
And we also get people to chair sessions as well, and that's that's quite daunting. But I think it's a great way of building your confidence and getting known to people as well. Actually,
You were saying you thought the halcyon days maybe of research is over. I must say I'm quite optimistic about certainly clinically in osteoporosis that we're just at the start of an upslope. When I look at other specialties.
Yeah.
20 years ago when I started in osteoporosis, it was very analog. You were either taking a bisphosphonate or you weren't taking a bisphosphonate, and that was it. And if you were really clever, you switched between alendronate and risedronate.
Yeah.
Whereas now we can choose anabolics, dual action treatments, anti-resorptives. We're thinking about really trying to make it personalised.
Yeah.
And I'm really, when I look at where other specialties are, I'm quite excited about the future.
No,
in
you're
bone.
absolutely right, David. I think I meant really the halcyon days in terms of the ease at which you could do things. But now, I mean, the technology is great now, isn't it? I mean, the omics revolution. So you're right. There's going to be a lot of exciting things happening. Personalised medicine, and we're identifying drug targets, and so there's going to be lots and lots of things happening. So it's exciting in that way, but maybe a little bit more difficult. I mean, the other thing is now you have to work in teams. You have to work together. Whereas I think again, when going back to my PhD, I remember one of the senior scientists in the lab who made some major discoveries in vitamin D metabolism. He worked completely on his own. Even washed did his own washing up in the lab. But now you need bioinformatics people. You need so many people in the team now.
That's why I see Richie surrounds himself with all these people who come from engineering, computer science, and builds a team from that.
Yeah. Well, the imaging techniques are just amazing now as well.
Yeah, I don't. My PhDs and postdocs, none of them come from bone.
Mm-hmm.
They're chemists, they're engineers, particularly mechanical engineers, and bone is quite good. As soon as you start talking about diseases and the impact that research can have, people from other fields really get on board. It's quite inspiring, I think.
Yeah.
We need to really use that as much as we possibly can.
Mm-hmm.
There's still so much to be discovered as well in bone. That's the other thing again, I think in other areas we're maybe a wee bit further on. I think in bone we're a few years behind some other areas. You still come to meetings like this and you hear about whole new pathways that are just being described, and you think this is very exciting. It's like my analogy the other day. We were talking, Richie and I said, you don't expect to sail around the South Atlantic and find undiscovered islands. But I think in bone research there still are undiscovered islands out there when you're sailing.
Well, I think the extraordinary thing is, I think in terms of molecular understanding, that's absolutely true. But even in terms of anatomy, because through studying alkaptonuria, this disease which causes this terrible osteoarthropathy, we worked with Alan Boyd, and we've actually identified some new microanatomical structures in bone that we didn't know existed before. We found them in this rare disease, and then we actually realised they're present in aging bone and they're present in osteoarthritis. So they're not disease specific. So even at the anatomical level,
That's amazing. We've been studying the human skeleton for 3,000 years, and you're finding new things.
Well, I mean, people have said I'm sure you've had these sort of comments. Oh, why are you interested in anatomy? Leonardo,
it's all been done.
But
Galen was there.
But it's not, And I mean, even today, some of the imaging sessions I've been to, well, not specific imaging, but people doing cell work and doing imaging. Then it's just amazing. I mean, to see I love the zebrafish stuff. If I was starting again, I'd definitely like to do some,
I had the same feeling as well, yeah.
Yeah.
The thing that strikes me is that a lot of this really good science, a lot of these new discoveries, the pathways, etc., they're being presented by PhD students and postdocs at the conferences. I mean, there's clearly a very talented crop of people who are very highly skilled and well supervised, I bet too. I mean, it's amazing.
Yeah. Despite the more difficult career pathways now, we've still got lots of really, really super young people in the field. Yeah.
And they will find a way.
Yeah, absolutely.
Thank you for talking to us.
Thanks very much, and I hope this series does inspire people, and I hope I've not been too negative by saying the halcyon days are planned. I mean, every era brings different challenges and different opportunities. So yeah,
absolutely, the conference has been a huge success, and congratulations to you and all the rest who worked on it.
Well, lots of people worked hard on it. Yeah.
Congratulations.
Thank you. Thanks.
Bye.
David, we've had a really wonderful second half of ECTS Live 2023. What were your takeaways from the second half of the conference?
Yeah, it's been great, Richie. I'm exhausted. I know colleagues at work sometimes think you go to conferences for a rest. Well, you and I can testify that if you're running a podcast at a conference, it's certainly not a rest. The things that I've brought away from the second half, more things about quality of bone, which we're always interested in. Things that I will go away and think about. We had that really interesting chat with Annalisa Schmidt about circadian rhythm and how sleep patterns affect bone. There's a lot of interest out there now about how sleep affects the cardiovascular system and the immune system. Guess what? It may actually affect bone health as well. So that's one I'll keep an eye out for in future. There were some interesting clinical stuff and some interesting clinical talks. Clinicians will be interested to hear there was a presentation about tapering the dose of denosumab and how patients respond to that, which, definitely watch this space on that. Some interesting stuff about giving IV bisphosphonate according to bone turnover markers rather than simply giving it every 12 months or every 18 months. And another interesting thing just about how you treat people who have fractured but who have normal bone mineral density, particularly people who have typical fractures like a hip fracture, but you arrange a DEXA scan and the bone density is normal. Now, you and I probably know there's a problem with the quality there, but just how you explain to patients that your bone density is normal, but I want to give you treatment to increase the density of the bone, or indeed, is it worth giving that treatment? And that's, again, a clinical sort of hot topic. And it's really interesting to hear these things discussed, to get views from all around the world. And I'm sure you'd agree with me. You come to a conference like this, you make notes, and then you go away and you have a think about the things that you've heard. And sometimes it takes days and weeks for these things to sink in, which is a useful reason, I suppose, as much as anything, for having this podcast where we can listen back to the things and remind ourselves about it. So your take, Sir Richie, you really enjoyed that interview with Jim Gallagher at the end, but what else stood out for you?
I think overall what stood out for me is the great diversity of people and research projects in bone right now. It seems like a really wide and interesting field, and that gives me great hope for the future. And it was really interesting to speak to Jim Gallagher. He was the head of department at Liverpool University when I did my PhD. He's kind of a giant in the field. He's done lots of really interesting work. He was really supportive to me when I was young, and I know many other people as well. It was great to hear from him his perspectives on how things have changed in research for the better or for the worse. And I think that was quite a nice round off to our podcast.
Yeah, it was good. It's been a really good few days.
It's been really fun, hasn't it? Listeners, I hope it comes across when you hear the interviews just how much fun we were having. And I think the guests were having a lot of fun with us as well. And overall, it really made me feel like we're doing a good thing with the podcast. If the people we're talking to are enjoying it and we're enjoying it, I think that's really important.
Absolutely. We had to edit out all the funniest bits, but you've got them in a reel somewhere.
We'll do an outtakes one day.
So, folks, hopefully we'll see you again soon with a more conventional episode with a guest. And until then, continue to look after your bones.
Bye-bye.
Bye now.